Okay, hi guys. We're going to do a lab today. Photosynthesis lab. So, get your notebooks out. I'm going to go through this on the video. I'm also going to post it as notes. I'm going to go through the whole video today, even though it says 2 parts, because I did it earlier, and anyway, never mind. So, Photosynthesis Lab. Our objective today is to discover what gases we breathe out and what gases a plant breathes out. How are we going to do that? Let me show you. I have this blue liquid. Well, actually, it looks greenish blue. It's called bromothymol blue. Now, instead of saying bromothymol blue all the time and even spelling that, we call it BTB for short. It's easier. Okay, now, if I take this BTB, so you can see it's green now, put it in a beaker, and then I'm going to take a straw and I'm going to blow into it. So what gas do we breathe out? First of all, let me explain what BTB is. BTB is an indicator. An indicator means it tells you when you have something. So BTB is an indicator for carbon dioxide. So in the presence of oxygen, BTB is bluish green. When there's carbon dioxide in the liquid, BTB turns yellow. Okay, so now, knowing that, I'm going to blow out into the BTB. Okay, so what color did it turn? Turned yellow. So if BTB turns yellow in the presence of carbon dioxide, then we must breathe out carbon dioxide, right? Okay, now we're going to see what color it turns if we put a plant in it. Let me make some more yellow BTB. Okay, so I have some BTB that's yellow, so that one's got carbon dioxide in it, and then I've got BTB that's bluish green, so this one has oxygen in it. All right, now, so our materials are going to be 2 test tubes. I just washed them so they're clean, with stoppers, the BTB solution, and then some green plant leaves. Now, it's a good thing I'm doing this because this green plant is about on its last leg. All right, so here's the procedure. So, blow into some BTB to turn it yellow. Okay, you just saw me do that. I did that right here to make it turn from blue to yellow. Done. Pour the BTB into a test tube marked 1. I didn't mark them. Okay. So I'm going to pour the yellow BTB into my test tube marked 1. Okay. And then I'm going to add 2 green leaves. Okay, try to find 2 decent green leaves. Okay, I'm going to add those to the test tube. Push them down in there. So they're down in the liquid. And then I'm going to stopper it. Okay, then pour some BTB into a different test tube marked number 2. Okay, so here's my other test tube marked 2. And into this one I'm going to put some blue BTB And then I'm going to take 2 leaves, put it into this test tube, find 2 decent green leaves. Okay. Once again, push them down in there. Okay. I'm going to leave those sit. Actually, I'm going to make it so I've got about the same amount of liquid in both. I've got 2 leaves. Got about the same amount of liquid in both. And I think I'm going to put some of the yellow. Yeah, actually, I'm just going to leave the yellow BTB out and the blue BTB out as well. And I was going to put it in a test tube, but I won't. And we will see if these change color. Okay, so hopefully you can see these. You might be able to see what's happening already. Yellow BTB with 2 green leaves, blue BTB with 2 green leaves. Okay, we're going to let that sit 24 hours. First, I also have a data table. So let's fill out this data table. Test tube, first of all, I'm going to say color of BTB at the beginning before I did anything to it was blue. After I blew into it, it turned yellow. Okay. Now, test tube 1 and test tube 2. Test tube 1 started out yellow, and test tube 2 started out blue. Okay, now I'm going to pause this video and let this sit and then come back to it. What do you think is going to happen? Make a guess. Hi guys. Okay, so I left the tubes here overnight. And can you see what happened? Here's tube 1. Remember, this was the one that was yellow, and I left it overnight, and now it's green. So what does that mean? This is the one that was blue. It's still blue. Okay, so this one definitely changed. So what does that mean? Okay, let's look at our data table and see if we can figure it out. All right. So test tube 1 was yellow. Now it's dark green. Test tube 2 was blue. It's still blue. Let's look at our beakers. Okay, this was the one that had the BTB in it. It's still dark bluish green. This was the one that I blew into. It was yellow, now it's greenish. It's definitely not yellow anymore. So what does that mean? All right, so if the tube is yellow, it's got carbon dioxide in it. If the tube is greenish or bluish, it's got oxygen. So I want you to think about what happened with this leaf that was in yellow BTB. So it had carbon dioxide in it, and now it's green. It's not yellow anymore. So what happened to the carbon dioxide that was in here, making the BTB yellow? Why did it turn colors? Why did it go from yellow to greenish blue? Okay, so there's our lab. There's your data. Now, your job is to write a conclusion paragraph using CER format. So that means you're going to give me a claim. You're going to support that claim with evidence. And you are also going to give me some reasoning. All right, so that's your assignment for today. There's our lab. I hope you enjoyed it. I wish you'd have been here to do it because it's more fun when you're here. I miss you guys. Bye.